Our next speaker is Nathalie Davoust.
Hi everyone, so my name is Nathalie Davoust and I'm going to talk to you about glial cells in a paraquat model of Parkinson's disease. So Parkinson's disease is the second most prevalent neurodegenerative disease after Alzheimer's, and it is associated with the selective loss of dopaminergic neurons in the substantia nigra pars compacta. The loss of dopaminergic neurons and the diminution of dopamine level into the brain will induce slowness of movement, resting tremors, and postural instability into Parkinson's patients. The major risk factors to develop Parkinson's disease is aging, but both genetic as well as environmental stress has been involved in Parkinson's disease. In Drosophila, both genetic and chemical models of Parkinson's disease have been developed. In this study, I'm going to use a paraquat-induced model of Parkinson. In this model, we are given paraquat by feeding the flies, and this induces an oxidative stress into the fly, production of radical oxygen species. And as you may know, dopaminergic neurons are very sensitive to oxidative stress, so there will be a dopaminergic neuron cell death, and plus, ultimately, the fly will die. So the question I wanted to ask with this project was, what are the mechanisms that may regulate dopaminergic neuron cell death, and what are the proteins in glial cells that could be involved in these processes? Indeed, in a human and also in rodent model of Parkinson's disease, it has been shown that glial cells, and in particular microglia, are able to synthesize factors which could be either deleterious or protective to neurons. For instance, microglia are able to sense danger signals, such as apoptotic corpus or extracellular ATP, and in an inflammasome-dependent manner, they will secrete pro-inflammatory cytokines, such as TNF-alpha, IL-1 beta, or even produce some ROS proteins. and this will in turn be deleterious for neuronal cells. On the other hand, we know that microglia, but also astrocytes, can produce neurotrophic factors, and this will be protective for the cells. So I use two approaches in parallel. First, a candidate gene screen in Drosophila, and then a meta-analysis of transcriptomic datasets from Parkinson's disease patients in order to identify new glial genes. So I will start with the result of the candidate gene screen. So I use an enhancer trap P-element strategy, about a bit more than 100 lines, and try to identify genes which would be expressed in adult glial cells. So here is a brain cryosection, and you can see a staining with the panglial marker RIPO, the pan-neuronal marker ILAVI, and genes of interest will be stained by LAGZ staining. So for the genes which were expressed in adult glial cells, I then did some functional assay with a paraquat treatment and look for genes which would affect paraquat sensitivity into the fly. As you can see here, these are two heterozygous loss-of-function mutant of spen, and you can see that the mutant lines are more sensitive to paraquat treatment, showing a pro-survival role of spen upon paraquat oxidative stress. In order to see what is the contribution of glial cells in this process, I use the pan-glial driver repo in order to shut down spen expression specifically in glial cells with a spen RNAi. So here is a qPCR showing the efficiency of the RNAi, and as you can see here, downregulation of spen expression in glial cell was sufficient to induce a more sensitive phenotype in flies. On the contrary, if I overexpress spen with a US spen construct, the flies were less sensitive to paraquat treatment. I also did some tubulin GAL80 system experiment in order to shut down spen expression only in adult glial cells, and still I have a significant difference in the survival curve, showing that glial inhibition of spen in adult flies still render flies more sensitive to paraquat. So, in conclusion of this first part, we identify with our Drosophila candidate gene screen spen as putative factors of sensitivity to paraquat. So a little bit of background to tell you what is spen. Spen is a member of a protein family, very well conserved along evolution. The orthologs of spen in human is sharp, and the ortholog of spen in mouse is mild. There are two functional domains, an RNA recognition motif and a SPOC spen paralog ortholog C-terminal domain motif. These two domains have been involved in different functions at the mRNA level, like controlling mRNA splicing, stability, and translation, but also spen has been associated as a transcription regulators. Pleiotropic function have been associated to spen, in particular during neuronal development, and in the early 20s, Chen and Ribe have shown that glia cell fate and axon guidance were controlled by spen. Last year, we have shown in the team that the survival of interomatidial cells during retina development was also controlled by spen. Spen is a highly connected protein involved in many processes, and as you can see here, it has been shown that spen is connected to the Notch pathway. It directly interacts with Notch, suppressor of hairless and hairless. So I started a collaboration with Laurent Senier in Lyon. Laurent is a specialist of Notch in glial cells. That was a good move for me. So here we use, in order to follow Notch activation, we use a Notch responsive element GFP construct. And here in this adult brain, you can see that in control condition, there is expression of Notch responsive element GFP, especially in the antennal lobe and in the mushroom body. And this expression has been shown to be glial. And if you do suppress spen expression using another driver, glial driver, EA81, you can see that there is a significant diminution of the Notch signaling in these glial cells. We further demonstrate the interaction between spen and Notch in adult glial cells by doing some epistasy experiments and expressing on the same chromosome both a UAS Notch intracellular domain and spen RNAi. So as expected, the expression of Notch intra induce Notch regulation, and once again, spen is able to downregulate this activation in glial cells. So this plus the literature data about interaction between spen and notch made us thinking that the spen is necessary to maintain notch signaling in adult glia. For the first part of my talk, I'm going now to talk to you a bit about the human results we've got. So we did that in collaboration with Laurent Pais and Serge Nataf. And what we did is a meta-analysis approach using a transcriptomic data set which were available. And so to include a Affymetrix whole genome microarray in our study, we decided to focus on studies which were specific to substantia nigra transcriptome, and also on studies which have both Parkinson and healthy patients in the same studies. So we obtained more than 3,000 probes which were differentially expressed between Parkinson and healthy patients. Then, using a G-profiler databases, we were able to find Drosophila orthologs. This would be, and then refreshing the data with GoTerm and FlyMine, we finally end up with 16 glial genes, and among these genes, we found spen. So, in conclusion, both using a Drosophila candidate gene screen and human transcriptome data analysis, we were able to identify spen, and we found that spen is expressed in adult glial cells in Drosophila brain. We also found that spen expression is modulated in Parkinson patients, but also upon paraquat treatment. I didn't show you that today, but we've got the data. And we found that spen was necessary to maintain notch activation in adult glial cells, that we do have a basal level of notch activation in the brain, and that spen was necessary for this. I would like to finish by say thanks to the people who work with me. First, Bertrand Mollereau, who is a team leader and who let me do my own project in his team. It's really nice. And the students who did the work, so Mathieu Kernet, who did the screen, the Drosophila screen, Valérie Goubard and Victor, who helped me with the notch part of the project, and Ryan as well. And thanks to my collaborators.
Great. Do we have any questions?
Yeah. So in the I was a question. Spen has many attractors, so the woman was asking me if we try other interactors to see if there was link over the notch, right? So in the pupil retina development, in fact, the effect of spen on survival of IOCs was due to EGFR signaling and not to the notch pathway signaling. But in the Drosophila adult brain, for the moment, we didn't try any other pathway yet. What is the consequence of loss of spen if I just knock it down as a spen RNAi? You mean with RipoGal4 or with another behavior? Well, we do have, in the paraquat model, we do have an effect on climbing, but if we do not treat the fly with paraquat, the fly looks normal. I mean, there is no climbing defect, or I didn't see anything. What we did also is looking at glial cell number, and our preliminary data show that there is no loss of glial cells upon spen downregulation. Thanks.